middle of it. And what I do is I tell people to move that dot till something seems to happen to that bar. All right, so back here I see the bar with the crosshash. I move in, and then something happens. The crosshash disappears into just a line. All right, so here we are back at our eye again. And let's start talking about sort of the most important part. This is the part of your eye, the retina. It's the part of your brain that is in your eye. Now, here is a picture, a schematic of a retina, okay? And first you'll notice there are these receptors called rods and cones, and we'll talk about those in a minute. And then those receptors connect to these things called horizontal and bipolar cells, which take the chemical output of the receptors and eventually turn them into stimulation for these ganglion cells, which are the first proper nerve, well, I shouldn't say proper, they're the nerve cells that actually produce firing spikes that we're used to for nerve cells. But when you look at this, you should look at that curvature, and it should bother you a little bit, right? So here's the picture of my eye. Okay, but you see, of course, my thing is bent this way, and this thing is sort of bent that way. So actually, what I need to do, and I'm really proud of this, is we need to rotate that picture so this curvature here is like this curvature there, at which point you realize what comes up this way? The light, all right? The human system has what's referred to as an inverted retina, okay? And that is the light comes in and passes through all of this gunk. It passes through, there are blood vessels in here, there's nerve cells, it passes right through them, then it hits your photoreceptors, okay? And then any stray light keeps going, sort of gets absorbed. This background layer, this is called the pigment epithelium. This is actually a very black pigment in the back of your eye. So you might ask, why would you build an inverted retina?
Why would you build an inverted retina?
Nobody asked me, but here's some speculation. There are a couple of things. The reason that I like best is, what happens is that the light goes through here, and anyone that happens to hit the photoreceptors right there gets absorbed, and then after that, it hits this pigment epithelium, okay? And what that means is the pigment epithelium absorbs any stray light. So it eliminates scatter within the eye. And that gives you a very sharp resolution. It reduces your ability to see at night a little bit. We'll talk about the sensitivity of rods and cones in a little bit. But it gives you this idea that, you know, first the light, whatever light gets absorbed by the detectors goes into processing, and everything else is absorbed and removed. It doesn't cause any disturbance. It does mean that there's a funny sort of thing that goes on. These structures produce, they're clear because they're made out of, you know, gelatinous stuff that is biological tissue. But of course it causes little shadows, right? So if you go right past the blood vessel, it'll be actually a little bit of a shadow. So you might ask, why don't we see these shadows?
Why don't we see these shadows?
The human eyeball is remarkable. Anything that's perfectly stabilized on your eye, the brain just removes. Okay, so that shadow stays exactly in the same place on the retina. The brain just removes it. In fact, there's some cool tricks you can do by building little stabilization systems that will keep something projected exactly the same place on the eye, and eventually you just don't see it. So anyway, so that's the inverted retina of the eye. Now, if you were to take a look a little more carefully at these photoreceptors and actually look at them for real, you would see two types. You would see rods and cones. Now, we've looked at this picture before when we were talking about color, but now we want to talk about the anatomy. So the rods are these little skinny structures here, and there are something like, it says 120 million rods plus or minus within your retina. They are very, very light sensitive, about 1,000 times more light sensitive than the cones, if given time to adapt. We'll see that in a minute. And it can sort of help you discriminate between different illumination levels at very low overall illumination. The other structures here are the cones, and we've actually seen these before. The cones are the things that allow you to discriminate color, and we talked about them when we talked about the short, medium, and long wavelength cones. The cones are also the ones that are packed within your, what are called your fovea, that is that middle area that has the very high resolution. We'll talk about the impact of that in a minute. And It's the cones that give you sort of what we tend to think of as the high quality vision, and the rods sort of provide everything else. Now, by the way, even if you didn't have an electron microscope, and even if you hadn't sort of peeled apart people's retinas, which sounds kind of gross, you would know that there are at least two kinds of systems going on in the eye. And one of the ways that this was discovered is in what's referred to as dark adaptation experiments, okay? So in dark adaptation, here's what we do. We put you in a dark room for months at a time. No. You put somebody in a dark room, and you, let's say after 1 minute, you say, how bright a light do I have to have a light to be before you can tell that it's on? Okay? So you do that. And then later we do it all over again, and we do it for 2 minutes. And then we do it for 3 minutes, right? So we track to see how much light you need in order to detect that there's some illumination. Now, we've all had this experience before, right? You walk in from a bright daylight into a dark room, and it takes a little while, we say, for your eyes to adjust. Well, that adjustment is not your pupil opening and closing. That's a rapid response. What's happening is that the photoreceptors in your eye are adjusting to the amount of light, all right? And if I were to track that, I would see this kind of a curve, okay? I would see a curve that starts going down like this and starts to look like it's going to flatten out. And then somewhere, this says about 8 minutes, varies on person to person, somewhere around there, all of a sudden it starts to drop down again, okay? What's happened is your rods are now kicking in. Your rods are now coming into play. And you can go way, much, much, much lower threshold, all right? And so, you know, when you have a single system, you never get psychophysics curves that have sharp breaks like that, right? And basically what you're seeing here is there are two curves. There's a curve here for cones and a curve here for rods, and this was where they intersect, all right? And this is sort of a well-known phenomena which implied that there was multiple systems within the retina in terms of light sensitivity. So let's take a little bit closer look at rods and cones. They have a certain similar structure. They have this, we'll call it an outer part, but remember, the pigment epithelium is up here and the light comes in that way. But this is called the outer part because it's this inverted retina thing, all right? And this inverted part is where you have these disc-shaped elements that react to light, and they react to different wavelengths of light. We'll talk about the different wavelengths in a minute. And those wavelengths and those reactions set up chemical reactions in the cell. And of course, it is still a cell, so it's got these inner elements, these organelles, and a cell nucleus. And then what's important is it has this synaptic body where it changes the chemical properties at the synapses depending upon the light. So let's take a look just at the rod for a minute. The rod has this chemical, these discs made out of what's called rhodopsin, all right? And here you can, this is an electron micrograph of what actually looks like. And this outer segment is made up of these plates, the rhodopsin plates. And what happens is that the rhodopsin is a chemical that when it gets hit by photons, it splits into two chemicals. I don't remember both their names. One of them actually is associated with vitamin A. Remember, your mother always told you had to eat your carrots in order to see well? Where does that come from? Well, the vitamin A in the carrots is involved in the chemical that's involved in rods being able to see light. So I'm presuming that that's why your mother told you that. Just ask her. All right. To stress the sort of chemical nature of what's going on, there's a slide here that just shows the same rod, and there's sort of this change in these sodium ions. And here I wrote it down in the notes that when the rhodopsin splits, one part's called opsin, and the other part is this vitamin A bit. And when light comes in, this change is sometimes referred to as bleaching the rods. Right, so they start to change more, and how bleached they are is sort of how adapted they are, right? So when they're not bleached at all, a very small amount of light now coming in will change it. After you bleach them a bit, now you need a different. You're now operating at a different light level. In fact, there were studies that were done that were showing that a single photon. So those of you familiar with the physics of light, a single photon can make a change in the cell that this system can detect, which is just remarkable, right? The ability of the system to detect small changes of illumination. And you can sort of ignore this diagram over here. It's just showing you that it's understood at a pretty detailed level how the light makes a change in the chemistry. and that the chemistry then changes what goes on in these synapses. Now, we said before that each of these photodetectors respond differentially to wavelengths of light. We showed you short, medium, and long. The one we didn't show you before was the one that includes rhodopsin. All right, so here's the blue or the short pigment, the green and the red, which we've looked at before. And here is what the rod responds to. And you see the rod is relatively broad spectrum, okay, which is good because it means that if there's any of this kind of wavelengths of light coming in at night, the rods will be able to see them even though they have a very, very low amount of illumination that they're sensitive to. We're not going to talk more about cones and the tri-stimulus theory of color because we did that last time, but these are the four operating spectral responses that each of the photodetectors give you. We did also look before about how the cones are not evenly distributed, right? There are many, many more red and green ones than there are blue ones. Remember we were talking about that? And we said that most of your color vision is based just upon the ratio between the red and the green, and the blue is just there to give you a little bit of sort of sensitivity to blue, but you don't really have high resolution in the blue channel. If you were to take a look in your eye, well, not your eye, because this has to be a dead eye, so some other eye, or your eye after you're dead. If you want to donate your eye when you're done, just send Megan a postcard. If you peeled the retina out and you stick it under electron micrograph, if you take a look at that, you'd see that as you moved along the eye, it was not the same everywhere. In the middle, which is the area referred to as the fovea, about 1.5 degrees, you see this magnificent hexagonal packing of photoreceptors. Okay? So hexagonal pack, so if you will, the pixels in the human eye are not on square grid, but they are on a grid. They're on a hexagonally packed grid. And the regularity of this is pretty remarkable, right? I mean, it gives you a very good sampling, a very dense and regular sampling of the underlying visual field, which is one of the reasons that you have such good high resolution up here and really lousy resolution out here. You see, so I'm looking straight at Megan. Wake up. Okay, good. So I'm looking at, I can't really tell how many fingers I'm holding up. I mean, I happen to know, of course, because they're my fingers, so I can tell there's 7 of them, but, or whatever. But I can't, if you do that yourself, just do it this way. If you want to have a partner, look them straight in the eye and have them bring their fingers in. And you'll see the fingers are there. You can have them wiggle them. But if they ask you how many fingers are they holding up, you can't really tell till it gets pretty close. And yet you feel like you've got all this high-resolution vision out here. That's an illusion that your brain is creating for you. Out here in your periphery, you just have good sensitivity to motion. In fact, wiggle your fingers, you'll see them right away. Why might you want to do that? Well, maybe you want to saccade your eyes over there. So the system is very good at detecting motion in the periphery and controlling the eye to look at it and foveating on what's there. But you actually only have high resolution in this very small area. In the periphery, as shown here, you have a few large cones. So the cones are actually big, and they're giving you overall color information. And then you have this high density, again, of rods. So with low levels of illumination, you can still see out really well. There, let's take a look at this picture. So this picture is degrees from center. So temporal is off to the side, nasal is towards the nose. And the first thing you'll see is that you've got all these cones that are mostly in the middle, in the fovea. That's exactly what we said. Notice, the same way we have rods out here in the periphery, notice you don't have any rods in your fovea. Oh, here's your blind spot, right? You don't have anything there. That's why it's blind. So what would one of the implications be of having no rods in your fovea? It would mean that you don't have very good low light perception in your fovea. And here's another experiment you can do. Some night, go outside when there's a bunch of stars, some that are really bright, some that are less bright. See if you can find a star that's less bright that stays there till you look at it. When you look at it directly, it disappears. And that's because your fovea can't see the low light that your rods do. So now let's get back to our retina. So here we are. So we've sort of talked, very briefly about the photodetectors. Now we're going to talk about the ganglion cells. One of the things about the ganglion cells is that they take information from these things called these horizontal and bipolar cells. And the horizontal cells, just as you might think, they take information across a local area and bring them together to the ganglion cells. So you might ask, why would you do that?
Why would you do that?
Thank you, Megan. Okay. There are a lot of reasons, one of which has to do with dynamic range.